Okay, so today we're going to continue on with the general theme of genetics. At least that's the ongoing theme here for the last chapter or so, and then on into the next one, I believe. So today then we're going to be looking at patterns of inheritance, or generally speaking, how living things are able to transfer characteristics from one generation to the next. And I mean, we know the mechanism of how is through DNA. Of course, we obviously haven't known that forever. We discovered that once we um, had microscopy, and so then that led us to see things we didn't know existed. But even before we really knew that there were things like DNA and chromosomes, and then that there are genes on chromosomes that code for characteristics of living things. We had to learn or study those concepts theoretically, and then apply research to those theories. And so Gregor Mendel, who is considered the father of genetics, or the first scientist to really do groundbreaking research in genetics, led us basically to the same concepts, principles that we apply today. He, among other things, experimented primarily with garden peas, just the type of pea that you grow in your garden to eat. And so he used thousands of plants that he bred, and bred certain characteristics, or I should say plants with certain characteristics, back and forth to each other, and that eventually led him to come up with, again, some of the fundamental concepts of genetics. So basically, he set the framework for the study of genetics, even on through today. He was breaking sort of a dogma, or a previous theory where most people believed that offspring got a blending of characteristics from their parents. Generally speaking, the idea would be something like, um, if you're breeding a plant that has red flowered, a red flowered plant with a white flowered plant, that the offspring would tend to be pink. Or that humans, you look at parents and you look at the kids of parents, and sometimes one child might have a certain characteristic, like maybe they have their dad's eye color, but they actually have their mom's hair color. Maybe they even have a cleft in their chin, and that was something that maybe came from a grandparent. But the idea that the offspring would not get a sort of a unique set, but a blended set of traits. Um, we also know now through the work of Mendel and others that there can actually be um, a couple of main ways that offspring do receive traits from their parents. They can either get it via what we call a continuous variation, which means there's a whole range of small differences. In other words, in humans, for example, you have everything from black, pure black hair, to almost white colored hair. And I don't mean like gray hair when you get older, but just individuals that have very, very, very light colored blonde hair. And again, pretty much everything in between. There are various shades of people with blonde hair, various shades of brown and red and blonde, and so forth. So there are those kinds of variations where traits can be a continuous range of options, even eye color. Not all people have the exact same blue colored eyes. But then there also is discontinuous variation. A white flower in a plant, for example, or maybe it has a violet colored flower, or maybe it has a red flower, but you don't see a continuous range of variation. Now, maybe you garden or you're thinking about this, well, what about roses? You know, there aren't just like red, white, you know, specific color roses. There are now almost a continuous range of variants in color of flowers. And that's because of the extensive breeding we've done to try and get all those different colors. But just be aware that there are kind of two ways that traits can show themselves, either in a continuous range or very specific, discrete examples. All right, so a couple of terms we have to go through, so we're all, again, on the same page with respect to understanding the terminology. So again, Gregor Mendel's process for testing characteristics, in other words, performing test crosses, basically involved hybridizations where, for example, he would breed a plant that always had violet colored flowers. So every time this plant bred with another violet colored plant, it would always make violet colored flowers. And so he would breed that type of plant with a true breeding white flowered plant. Again, one that every time you bred this white plant to another white flowered plant, it always made white flowers. So that's kind of a fundamental part of his process, is starting out with true breeding individuals. And so this first generation of these parents would be the P generation, or the parental generation. Once those individuals were bred to each other, so again, the violet producing plant to the white flower producing plant, you would then look at the F1 generation, the first filial, but again, basically just the first generation of offspring from those two parents. Then what would happen is the offspring of that first generation would be self-fertilized, basically. So again, you have all of these offspring, and in this case, notice they have the purple colored flowers. In this case, breeding a true violet producing plant and a true white flower producing plant, you only ever get purple flowers. At least that's what he found in his experimentation. And so then self-fertilizing individuals from this generation to breed those together to produce the F2 generation, the second filial generation. Now he started finding that not only did he get individuals with purple flowers, but he also was getting some individuals that had the white flowers from back in this parental generation. But what was really kind of fundamental to his research, that he did these kinds of test crosses over and over and over again. Remember, it wasn't just one plant, you know, with one plant and then crossbreed those plants, self-fertilize them, and then get offspring. He did this hundreds and hundreds and hundreds of times. And so it's really these ratios that he came up with that became the basis of his work. And notice that on average, for every 1,000 offspring, about 3/4 of them were always violet colored flowers, and about 1/4 were the white colored flowers. Again, notice it's not exactly 750 to 250, but it's pretty close to a ratio of 3 violet colored flowers to every 1 white colored flower. So if we jump down below here, we're back at looking at the ratio there. So again, even though it wasn't a perfect ratio, it was a pretty close to a 3-to-1 ratio of these violet colored parent flowers to the white colored flowers. So what we're looking at here again are traits. traits being the variations of physical appearance in a heritable trait, in a characteristic that can be inherited by offspring or passed along from parents to offspring. So again, notice in this cross of true breeding individuals that, again, all of the F1 generation were violet colored. And remember that if we believed what a lot of people believed back then, that if you breed a violet colored plant to a white colored plant, that you would have actually predicted that there would have been some sort of intermediate color, like a pale violet color, or maybe even an equal proportion of violet and white, breeding these two individuals together. But again, what happened instead is that 100% of that F1 generation were violet colored. And you could have bred these 1,000,000 times, and 1,000,000 times you would have only got plants that had violet colored flowers. Now again, prior to Mendel, you would have predicted that this would have been a blending of characteristics if what we believed before Mendel was true. And again, indeed, Mendel found that was not the case. So again, Mendel used pea plants in his experimentation. And it wasn't just the color of the plants. It was a number of different characteristics. He looked at the shape of the seed, whether it was round or wrinkled. He looked at the seed color, yellow and green seeds, or peas in other words. Of course, the flower color like we just talked about. Also the pod shape. There are those that were considered to be inflated. The pea pod looked puffy, versus those that were more constricted, where you could see, where exactly the individual peas were inside of that pod. Also the color of the pod, color of the seed, but also the color of the pod. And then a few other specific things about the flower, where the flower was located. Was it down here in the axial position, coming off more the lower part of the stem, or were the flowers coming off the more terminal end of the stem, near the top? And then finally the height, either relatively tall plants or much shorter plants. So again, he didn't do this experiment just with one characteristic, because you could say, well, if it was just flower color, maybe that's just how the flower color is. But again, he did that experiment with a lot of other characteristics, still using the pea plant initially, but a lot of different characteristics. And so again, what he always got, I keep looking down so I can find my little hotkeys to get my pen here, is that again, if you do a test cross of a pure yellow producing seed plant, and you cross that with a plant that only produces green seeds when you cross them to each other, in this case, what he got was, again, in the F1 generation, 100% of the offspring were yellow. And so we refer to that as the phenotype, how something looks. So the phenotype of the F1 generation was yellow. And not only yellow, but 100% yellow. And then again, remember, you would always self-fertilize the F1 generation. So breed any one of these individuals back to any one of the other individuals in the F1 generation. And then in the F2 generation, again, same thing. About 75% of them produced yellow seeds, and only about 1 in 4, or 25%, produced the green seeds. So again, whether we're talking flower color or seed color or the other characteristics, when he did this type of test crossing, he always got that same ratio in the F2 generation of 3:1. So again, the phenotypes are the physical expression of the trait, the way something looks, as transmitted by the alleles. And we'll talk about what those are in a minute. The genotype is basically what the genetic makeup is. So not how it looks, but genetically what it is. And this is where the alleles come in. So in this test cross, we're doing the exact same test cross we did here. We're just representing these colors now by the alleles. And I do need to point out there is an error here. This is actually the genotype for yellow. For some reason, this image got swapped. So when we're talking about dominant alleles, which we know in this case is whichever one makes yellow, because again, that's 100% of our offspring are yellow in this first test cross. So we actually can do a test cross of two characteristics, and whatever the offspring are that show up in the F1 generation are ultimately going to tell us that those are the dominant alleles or the dominant characteristics. And then we represent those by all capital letters. So if we know yellow is dominant because it's what the 100% of the offspring are in the F1, then we represent that by two uppercase. In this case, we can choose anything but yellow, Y for yellow. And again, sorry, these for some reason on this image got swapped. So two uppercase letters for dominant alleles, two lowercase letters for what we refer to then as the recessive allele. Now there are a couple other things that can happen. There can be codominance and so forth, but typically we have a dominant characteristic and a recessive characteristic. And so again, we represent those by uppercase for the dominant, lowercase for the recessive. And so we represent then the same cross here, a yellow P times a green P. We do the test cross, we get 100% of the offspring. And we'll look at in a minute what the offspring, how we come across this genotype. So we'll look at that in a minute. And then when we self-fertilize those, again, we still get a combined 75% that are yellow and 25% that are green. It's the exact same thing on this side. The only difference is now, instead of just having yellow and green, we actually have three possible genotypes. When we combine uppercase dominant alleles with lowercase recessive alleles, we can actually get three different combinations of genotypes. And basically this whole process is the law of dominance. Being able to do test crosses which result in an F1 generation that tells us what the dominant characteristic is. All right. For a dominant characteristic to show itself, I'll go back here, we only need one dominant allele to be present. Notice all of these F1 generation offspring have a genotype with one uppercase and one lowercase y, a dominant allele and a recessive allele. And again, that's all of the offspring between that test cross. But yet we get offspring that have three different combinations, not just that combination. But now, again, we have offspring that can have both uppercase y's, in other words, dominant, both lowercase y's, in other words, recessive. But then we can also have a mixture of the two like we did in our F1 generation. And as long as one dominant allele is present, like it is in both of these, then the dominant characteristic is going to express itself, in this case, the yellow color. Well, what happens then if both recessive alleles are present? Well, go back in. In the example we just saw, the only way we can get a green P is if we have both recessive alleles present. And we're always going to get the dominant color if at least one dominant allele is present. In recessive characteristics, for example, in humans, you generally would need both parents to be carrying a recessive allele for a characteristic, whatever that characteristic is, for the child to have a chance to get it. Now again, remember, just because the mother and the father both have a recessive allele, like might be the case with this parent, right? Any two of these that we cross to each other, both of them have a recessive allele. And then again, we know that about 1 out of every 4 offspring carry both recessive alleles, which is the only way you get a green colored pea. In the case of albinism in humans, and again, we mean true albinos, not just people with fair skin, but basically people that lack pigment. There is no pigment in their eyes, they don't have pigment in their skin. The only way that an offspring can get the characteristic of being an albino is if both parents carry the recessive allele. But also remember, just because they do, that doesn't mean that any given child they have is going to be an albino in this case. It would be about, again, a 1 out of every 4 chance. So does that mean that if this mother and the same father had 4 children, that one of them would have to be an albino? So if you think about that for a second, the answer is actually no. Now we start getting into probability, which maybe some of you have had that in statistics, but just because there's a 1 in 4 chance doesn't mean that every 4th child is still going to carry the characteristics. And I'm exaggerating just to make a point. 2 parents could have 100 children, which I know isn't physically possible, but they could have 100 children and not one of them might end up being albino. Or it could conversely be that a higher percentage of them are albinos than you would predict. Again, probability plays in there as well and can somewhat confuse people, but statistics is definitely a class worth taking, if for no other reason, to make sure you kind of understand numbers and how they can be manipulated. So remember, what's really happening here when we breed these individuals and we get offspring is that, again, during that process of meiosis, in this case, remember, we're in meiosis 1 right now, the law of segregation states that the genes must segregate equally into the gametes. Remember, these daughter cells that are produced, the daughter cells that are produced from the process of meiosis 1 are, again, identical genetic copies of each other. So they have equal opportunities of inheriting genes, basically. These genes are segregated equally into the gametes, and again, you're basically giving either one of these daughter cells an equal chance of inheriting any given gene that's found in the parents. A couple other characteristics to point out here, or a couple other terminology, I guess, is that performing a test cross, if we have, for example, parents, and we know what they look like, right? We know what their phenotype is. Again, I can see, well, this is a plant that produces yellow peas. Here's a plant that produces green peas. Again, remember, we have the foresight or the hindsight of having all of this stuff already understood for us. When Mendel was doing this, he didn't know what the results were going to be. He didn't even know really that there were these things that were microscopic that were controlling the characteristics. He didn't understand or didn't know about mitosis and meiosis. He didn't know how that worked. So he was just going off of what he observed. And so what he would do then is perform a test cross to determine what characteristic is present and whether it's a dominant trait like we talked about or a recessive trait. And again, by looking at it. Also, though, whether it was homozygous or heterozygous as far as the genotype. So again, we can perform a test cross to figure out what's going on here. Now again, imagine for the purpose of this experiment, we don't know anything about the genotype. All we do know is yellow and green. So if we breed the individual that's yellow and test cross it with an individual that's green, and again, in this case, if we've got pure breeding yellow plants and pure breeding green plants, again, remember that means anytime we breed one of these yellows to another yellow, it makes a yellow 100% of the time. And anytime we breed one of these greens to a green, it makes a green 100% of the time. So we breed those together, yellow mated with the green, and again, we already saw that we get 100% of our offspring are yellow. Now looking at the genotype, we know that if an individual is showing, so we've already done these test crosses and we understand dominant versus recessive. We figured out already from our previous test crosses that green individuals are recessive, right? And the yellow individuals are dominant. But remember on the previous slide as well, we saw that there are more than one genotype, right? You can have two uppercase Y's, or you can have an uppercase and a lowercase allele. And so remember, because this individual looks yellow, we don't necessarily know which of these two genotypes are present. And so the way we can figure out what is present is by doing a test cross. So again, in this case, all of our offspring are yellow. We know then that the only option for the other allele here is that it's an uppercase, a dominant allele like we had here. And so this kind of brings up this Punnett square here. We write the alleles in of the parents, and then we simply bring it down and bring it across. Bring the uppercase down and the lowercase across. Uppercase down, lowercase across, uppercase down, lowercase across. And that gives us all of those possible combinations. Clear that out. So again, that leads us to understand that when we have these offspring, and we already know that this one has to be two lower cases, again, because it's a recessive characteristic, that's the only way it can be. But again, when it's still a dominant looking individual, it could have two uppercase alleles or one of each. And if we only get dominant colored offspring, then we know, again, the only possible way that can be is if this unknown genotype is uppercase. In other words, two dominant alleles because of pairing these up. So we do this again. Again, we have an individual that has yellow color, and we cross it with an individual that has green color. Again, we know what the alleles are for the green individual. And because the other individual is dominant, we know one of the alleles has to be a dominant allele. So we breed these individuals together, and now we end up getting about a 1-to-1 ratio of yellow offspring and green offspring, unlike the 100% yellow. So again, in this case, we know that the other allele of the parent has to be a recessive allele. And that's how, and the only way, we can get a 1-to-1 ratio of dominant phenotypes, yellow, to one recessive phenotype, which in this case is green. And so then that brings us back to these terms, homozygous, heterozygous. When we're referring to the allele combinations, if they're the same, in other words, if they're both uppercase dominant alleles, or if they're both recessive alleles, they're the same. And again, the Latin kind of root word of homo means similar, right? Same, one. And when we have combinations of dominant and recessive alleles in one offspring, then we refer to that combination as being heterozygous, meaning different or two. So homozygous would be both dominant or both recessive alleles, and heterozygous obviously can only be in reference to one of each, one dominant and one recessive allele. So again, lots of excuse me, lots of terminology. All right, so then let's look at this idea of a Punnett square again a little bit more. So basically, we use these diagrams to represent crosses between parental generation and then the F1 and F2 generation. And so we use these just as a way to kind of visually understand what's going on with respect to the genotypes and the phenotypes. Keep in mind that Mendel didn't use Punnett squares. He actually used matrix algebra. He used math, basically, rather than this more simplistic way of showing it. And again, remember, he didn't really know there were genes and alleles. He predicted that there were things that had to be controlling these characteristics that he couldn't see or didn't identify. So he predicted there had to be something going on that he couldn't see, which makes it even more interesting that he was able to visualize some of these things. So again, we're looking at exactly the same thing we've looked at on the previous slides. So in this case, we're just doing a monohybrid cross. Again, mono being another root word that means one. We're just picking one characteristic. Yellow peas versus green peas. In other words, color, right? The color of the pea, one characteristic. And again, picking pure breeding individuals, always make yellow, always make green. And so we know then the allele combinations have to be what they're shown here. The dominant have to be homozygous, dominant. The green peas have to be homozygous, recessive, both lowercase. And so then remember what's happening here is meiosis, right? You have an individual parent, like this green colored plant, is giving one of its alleles and then the other allele separately to the offspring. The other parent, again, the same thing, giving one of its alleles and the other allele separately to produce an offspring that has the correct number of alleles, which is, again, 2 in this case. And again, nothing new here. It's exactly the same thing we've been talking about. We get a heterozygous dominant offspring, right? We then self-pollinate these heterozygous dominant offspring from the F1 generation, and we split their alleles out, right? That parent gives one of those and one of those, and then another individual gives that allele and that allele. So again, there's more than one of these individuals that are each giving 2 of their alleles to their potential offspring. And then again, basically we have the Punnett square here at the bottom, where we have the possible alleles from the parents, and then all we're doing again is bringing each one over and down for all of the offspring. And again, we can see the phenotypes. It's getting a little messy, isn't it? Let me erase that. You see the phenotypes again are 3 yellow for every 1 green. So this gives us a phenotypic ratio of 3 yellow to 1 green. But it also gives us a genotypic ratio of 1 homozygous dominant, that's this individual, for every 2 heterozygous dominant individuals, and that would be these 2, for every 1 homozygous recessive individual, which again is that offspring. So we've said the same thing several times now, just to illustrate, I guess more than anything, all of the terminology that comes into play when we're talking about Mendelian genetics. Now again, those were just monohybrid crosses, one characteristic. Color of the pea pod, color of the pea, color of the flower. Mendel also conducted dihybrid crosses, which then takes into account 2 characteristics at the same time. So in this case, he was breeding individuals that had yellow-colored peas, and also the other characteristic was round versus shriveled. So yellow and round are both dominant characteristics. Green and wrinkled, I guess I said shriveled, but wrinkled, are both recessive characteristics. And so you can see that our original Punnett square was just 4 squares, right? Because we only had 2 characteristics, well, 1 characteristic, 2 parents, but 1 characteristic, a monohybrid cross. But now that we've got 2 characteristics, a dihybrid cross, our Punnett square jumps up to basically 16 combinations. And so it gets a little bit messier, just making sure you can track these genes. And I would really suggest doing some of these Punnett squares as practice. Just even if it's like redo this one, you know what the parents are, so you know how to breed them, how to mix up the possible combinations of alleles to get the first generation. And so as long as you know what the alleles are, now you just need to be able to sort out how to segregate those gametes to the possible combinations. Now remember, the options need to have 2 alleles, right? They have to have an allele that relates to the color, yellow or green. In this case, remember, those are the Y's, upper or lowercase y's. And then they also have to have the gamete that relates to the smooth or wrinkled, which are the R's in this case, the R's. And so it's just a matter of looking at these and thinking about all the combinations, right? You can have 2 uppercase alleles. You can have 1 upper and 1 lowercase allele. You can have a lowercase allele and an uppercase allele, like we have there. Or you could have a lowercase allele and another lowercase allele, like we have there. So for dihybrid crosses, those are going to be the 4 possible ways the alleles can be segregated. And then again, notice we're just repeating the same combinations for the other parent. Again, remember, we're breeding 2 of these parents together, so they're going to have the exact same genotypes. And in this case, when we're doing a dihybrid cross, our offspring have 4 possible phenotypes, right? They can be yellow, which we know is dominant. And so we've got two possible combinations for yellow-looking phenotypes. And then we also know the other option can be green or a wrinkled texture. So we have the dominant characteristics being yellow and being smooth, like this one is, right? Like both of these are. Or we can have the recessive characteristic being green or being wrinkled. And notice what happens now is we always get a ratio of 9 where the phenotype are both of the dominant characteristics, yellow and round, or smooth, whichever way you want to refer to that. And then we get 3 individuals that have one of the dominant characteristics being smooth and one of the recessive characteristics, in this case being green. We have 3 individuals that have the other possible combination, dominant being yellow, wrinkled being recessive, and then only 1 out of these 16 individuals that gets both recessive alleles. In other words, it's going to be green and it's going to have a wrinkled skin to it. And again, we can do this Punnett square, and again, all we're doing are bringing the alleles across. And then our offspring have 4 alleles again, just like both of our parents did. And again, we know if there's a dominant allele present, just one, if at least one, if there's a dominant allele present, the phenotype is going to show the dominant characteristic. And so all of these have one dominant allele for both the yellow color and the round shape. So again, you can count that out, and there are 9 of those. In this case, each of these have just one dominant allele for the smooth or round shape. So they're still round, but they carry both recessive alleles for the color, which is why these are all green. And then the other 3, kind of the same thing. They're carrying one dominant allele for the color in this case. So remember, yellow is dominant, but they're carrying both recessive alleles for the wrinkledness. And then, of course, again, the only reason this one is green and wrinkled is because it's carrying both recessive alleles. So, again, Mendel did these kinds of test crosses as well, and he always got a ratio of 9 to 3 to 3 to 1, no matter how many times he did that. And so the phenotypic ratio always came out to this when he was doing a dihybrid cross. And again, basically this random segregation that happens during the first meiotic division can lead to all of these possible combinations of the arrangements of the genes as they're transferred from the parent to the offspring. Now, remember, we talked about traits, can be continuous or discontinuous. We also have been talking about dominance versus recessivity. And there actually are a couple other ways that alleles can be expressed in offspring. There are certain living things, particularly in plants, where we can see what's called incomplete dominance. Incomplete dominance. And your book uses the example of snapdragons, where there are individuals that produce red-colored flowers. There are individuals that produce white-colored flowers. Like with peas, right? You could have violet or white. The difference is, though, that in this case, in snapdragons, there are actually instances where the offspring are pink, not red or not white, but somewhere in the middle, which would kind of lead to people back in Mendel's time going, see, we're right, the blended characteristics is how, characteristics are passed from parents to offspring. And again, in some cases, that's almost what's happening, but it's really about there being a lack of complete dominance. In other words, in the case of the snapdragons, we could represent R with these two uppercase alleles. Uppercase, again, referring to it being dominant. So if there are two uppercase alleles present, in the offspring are red colored flowers. And then if these alleles are present, the offspring are white. Now we could, in other examples, think these must be recessive alleles, lowercase alleles. But in experimenting with these, what's really going on is these are also dominant alleles. So for a plant to make a white offspring, the offspring has to get both dominant alleles for white. And what happens when you breed, say, these two individuals together, you're going to get a combination of the two mixtures of dominant alleles. So then what happens if you have an offspring with a R and a W allele? Well, in this case, again, you get a third color. You don't get white, you don't get red, you get a third color, which in this case happens to be pink. And so through experimentation again, basically this is how we came to understand that in some situations we actually have an incomplete dominance. If red were truly the dominant allele, then if it's present in the offspring should be red. Or if white were the dominant color, if the dominant W allele were present, then the offspring should be white, but they're pink. So that's how we know that this is actually an incomplete dominance. Neither one of these alleles are truly completely dominant alleles. Another interesting way to illustrate this is with looking at blood type. It's exactly the same thing we've been talking about, but it allows us to look at a different situation. We had incomplete dominance in snapdragons. In the human realm, with respect to blood types, we actually have what's called codominance, where both alleles, if they're together, can express themselves both equally. I remember in the previous slide, if both alleles were present, then we have a whole different color than red or white. In the realm of blood types, we can have a situation where we have, and you probably are aware of blood types typically, you can have an A allele or a B allele. And if you have two parents that have each one A and one B allele, when you again put the possible combinations together, you end up with about 1 out of every 4 offspring. So again, 1 out of every 4 offspring that's going to have genotype AA, which would be type A blood. On average, you're going to have about 2 offspring that have an A and a B allele. And then we, again, call that type AB. So about every 2 out of 4 offspring are going to have type AB. And then if the offspring got both B alleles, then we call that type B blood, which is also about 1 out of every 4 offspring. So a 1 to 2 to 1 ratio of genotypes A, AB, and B. So if you know anything about blood, or you don't fit into this, you know there's basically one more blood type, right? Type O. That fits in a slightly different way. But again, that's an example of codominance, where if it's only A alleles, then it's only type A blood. If it's only B alleles, it's only type B blood. But if both of them are present, they both express themselves. They are codominant. So you have a whole new blood type that's AB, basically. It's both A and B. But as I said, don't forget, and if you're this type, then you obviously would know that there's also type O blood. And so this comes into play in a slightly different way. Let me see here. I actually wanted to. So here's kind of the breakdown of the A and the B, and then this this sort of lac here. This I is, I guess, maybe think of it as a placeholder. It's not an A or a B. And so what happens, as long as one of these alleles are present, right? If A is present in any shape or form, the blood type is A. This parent would donate an A allele. This one basically is donating nothing. I mean, it's something, but for now, just think of it as nothing for a minute. And so that means this offspring is type A. Again, this offspring is getting an A allele from one parent and a B from the other parent. So that's a type AB blood. That'd be the same for this other one. To be a type B blood, you need both B alleles from your parent or a B allele from one parent. And then again, this sort of placeholder, you're still type B in that case. The only way that you can have type O blood is if you don't get an A allele or a B allele from either one of your parents. So again, 1 out of every 9 of these offspring, on average, wouldn't get an A or a B allele, so they end up being the type O blood. Now, again, I don't want to get into this too much. You can read the slide. But you may also know that you can have A positive, B negative. You can have positive and negative factors as well. And that's related to this thing called a rhesus factor, an Rh factor. It's an antigen that exists on the red blood cells in most people. And if you have the factor, you're considered to be a positive blood type. So again, you can be A positive, B positive, whatever. If you don't have that antigen, then you're considered to be a negative blood type, like O negative, AB negative. Most people have that positive factor, that antigen. About 85% of humans have that. So if you're a negative blood factor, you're fairly rare as things go. And then, there are 2 alleles or genetic variants of that antigen. Basically, you can think of it as a dominant and a recessive. Person who's negative has both recessive alleles. And if you're a positive, then you can have at least one of these. It can be dominant, like this, or dominant recessive. But as long as you have at least one dominant allele, you have a positive factor. And if you have 2 recessives, then you're going to have a negative blood factor. And again, these are also characteristics that are inherited from your parents. Also, just a little side note, somebody had asked this question one year, and I didn't know the information off the top of my head, so I went back and kind of added it in just out of interest. It used to be that before 2 people could get married, they had to have a blood test. And at the time, it was to check basically for this Rh type. During pregnancy, an Rh positive fetus, if it happens to be developing inside a woman who is an Rh negative, then that woman runs the risk of developing a certain disease, a hemolytic disease, in the newborn. So only women with Rh negative are at risk because, again, there's a chance, it's a bit of a random chance, I guess, but there's a chance that you could have a problem during pregnancy or have a child that would be born with a problem. And so basically the blood test would be testing for this rhesus factor. So if you were an Rh negative woman and to have an Rh positive child, father has to be Rh positive. Because again, as long as that positive is there, that means you're either both dominant alleles for the positive factor or you're heterozygous for the positive factor. But either way, if you do a Punnett square for either of these options, there's a 100% chance if the father is homozygous dominant, there's a 100% chance the child will be positive, which means no problems. But if the father is heterozygous for the Rh factor, then there's basically a 50-50 chance that the father could donate the dominant allele to the mother, to the child, I should say, which would be a positive. But if it donated the recessive allele, then there's a chance that the child could be Rh negative. And so again, there tends to be problems if that enters the blood, and it can cause a lot of problems. So it used to be that that they required a blood test before you could get married, and it was to see where the mother and the father stood with respect to this factor. And then potentially there could be some challenges if those two parents chose to have offspring. So Mendel mostly focused on plants, pea plants, for example, but we also have done a lot of experimentation in animals as well. And typically we focus on animals that can breed rapidly and make lots of offspring, but also species that have various characteristics. So the Drosophila, or the fruit fly, is one of the standard sort of experimental animals for genetics research. We don't use them in this class, simply lack of time and everything, but other biology classes typically would actually either breed these themselves or buy them from a biological supply place, and then be able to study the various genes and things, the different crosses and so forth. One of the things that we can look for in animals, and specifically we use the Drosophila for, is eye color. And that's because in Drosophila, the eye color is actually located on the X chromosome. And that means that it's an X-linked or sex-linked characteristic. Red eye color is the wild type, the sort of normal type, and it's the dominant color to white. So this individual has the dominant red eye, whereas this individual has the recessive eye color. And again, rather than the characteristic being carried anywhere else on the chromosomes, it's only, in this case, on the X chromosome, which again is meaning it's a sex-linked or X chromosome-linked characteristic. And so there are lots of experiments that we can do to understand how sex-linked characteristics can be transferred from parents to offspring. So this even muddies up our Punnett square a little bit, because even though we're only looking at one characteristic, eye color, we now have to make sure we also are taking into account sex, male versus female. So we breed a male fruit fly to a female fruit fly, a male that happens to have a red eye to a female that happens to have a white eye. So we not only have to carry the X and Y chromosomes, X and Y for male, 2 X chromosomes for a female, but we also have to carry the alleles for the characteristic, in this case eye color. So an uppercase W there on the X chromosome for the red eye, 2 recessive alleles on both X chromosomes for the white eye color. And then when we breed these parents together, it shows us what the offspring look like, not only their eye color, but the sex of the offspring. So we talk a little bit about this in the bio lab for this class, and of course the book covers it a little bit. But what I really want you to focus on for the purpose of understanding this chapter is really focus on understanding the terminology, what we mean by hybridizing, the terminology for the generations, P, F1, F2, the ratios that we get in the generations from true breeding individuals, the 3-to-1 ratio of dominant to recessive characteristics that we find. Definitely need to understand how to do a Punnett square. Again, whether you're given the alleles to just put them together, or even if you're given an unknown allele combination, but if you've got the offspring, then you can go backwards and figure out which allele is missing to understand what the genotype is of the parent. And again, more terminology, homozygous versus heterozygous. And again, realize it's not just about monohybrid crosses. We very rarely only have one discrete characteristic that we're interested in studying. Also remember, you can have incomplete dominance, which could result in individuals of a totally different characteristic if each of the dominant alleles are present in an offspring. If they're both the same alleles, you'll have one color. Both of the other dominant alleles will result in a different color, but if you have one of each, incomplete dominance, you get a third color option. And that, for example, our human blood system is an example of codominance, where you can have both A alleles making A, both B alleles making B, but if you now have one of each, you now have a totally new blood type, AB, where both alleles are expressing their dominance. Now, just real quickly, you probably are thinking, well, how are codominant and incomplete dominance really different? Because you have one option, a second option, and a third option. One option, a second option, and a third option. Remember, the difference in incomplete is that the third option is neither red nor white. It's actually something in between those two. It literally is kind of like mixing red and white and getting pink. Whereas in this case, it's like mixing A and B and getting A and B, not something that's in between. You have A and B, not somewhere in between. So that's kind of the difference between incomplete and codominance. And then the very last little bit of your book just shows you again in an animal where you are looking at a couple of different characteristics, that you can actually have another interesting situation that's called epistasis, where the presence of a particular gene ends up hiding or masking the expression of another. And so this is actually where you can generate albinos or albinism. Again, notice whenever there's a dominant allele present, you have one characteristic, but when you get the gene C present, it can actually mask the expression of A, and so rather than having a certain coat color, this kind of one called agu, or agouti, however you want to pronounce that, it actually masks that color and then just allows the individual to be an albino. So you can have a gene for a color of a coat in something like a mouse, a gene to make it brown colored, or a gene to make it black colored. But then you can also have another gene that, if it's present, can actually hide the coat color. And so again, that's called epistasis, the way we go about having that expression of a recessive characteristic, or basically masking the ability to express a normal coat color. All right, so again, with this section, lots of terminology. Just make sure you take the time to go through the slides, of course, but also then go back through the terminology. Make sure you look at the questions at the end of the chapters. And I really can't sort of express enough how important I think it is that you do practice some of these Punnett squares. We're going to do them in lab, in lab class, if you have the lab for this class. But again, just doing some of these practice problems, and there are tons of them online. You can just type in monohybrid Punnett square examples, and get a whole bunch of them that you could do to work on, or at least a few. It's kind of like doing math homework. It sucks, but if you don't do it, you're never really going to understand it or get any good at it. So take some time to do a few of those practice Punnett squares. All right, see you next time.